might be useful. Okay, so I didn't write down your names. Sorry about that. So I'm gonna read aloud some parts of your question. So one of the questions was, in one of the first classes, we said that people can show sign of Broca's aphasia even if there are no lesions to Broca's area. Okay. Now my question is, how is this possible and how it is explained? Another thought was that could it be possible that, along with the idea of brain plasticity, the different parts of the brains could change their function over the course of the lifetime of an individual? Very interesting questions. So the first one, why can you have Broca's aphasia if there are no lesions to Broca's area? So the stuff we are talking about, it might be an example of this, right? So there are two ways why a certain area might suffer from damage to other areas. So one is if you have a damage to the white matter surrounding this area, or bringing projections from other areas to this area, right? So usually this is called axonal damage or white matter damage or disconnection damage, right? And the reason why you have that is quite clear, because in order for a certain circuit or a certain area to work, it needs to get the information from the other areas it receives the information to, and also it needs to send off the output of its processing to the areas that are that require that output in order for the whole function to work. So simple example, Broca's area, right, might receive information, for instance, from your insular cortex, which triggers from your, let's say, executive system, the information about initiating the speech, and it re-triggers the information to your Broca's area. And the Broca's area is gonna, say, get the motor program for articulating words and transmitting it to your premotor cortex, which will retransmit the information to your motor cortex and finally move your lips, right? If any part of this circuit is damaged, then what you're gonna get is probably a deficit at the articulation of words, or a deficit at the initiation of the speech, right? So something that could look like an expressive aphasia, but in fact, the problem might be at some lower level. So for instance, premotor cortex damage, or some higher level, like again, at the insula that is transmitting the information of initiating the speech to the Broca's area. Okay. I mean, I'm not 100% sure that this is exactly what the insula does, but these are some hypotheses that people came up with. Okay. So this tells you that you might have it like the neuronal damage or the neuronal disruption at different levels, but then it might come out with the same kind of deficit, right? And then again, that's also a source of a lot of debate in the literature, because then leads to some people saying what really matters is the network itself, is not the specific area. And then some people bring this argument even further, and they say the single area is actually not doing what people claim it does. It is just part of a larger network, and the disruption of the area causes the disruption of the network. So you see this is kind of the mirror image of this argument. So the original question is, why do we get Broca's aphasia if Broca's area is intact? And the answer is, you might have a damage to the circuit, so a damage to the white matter or damage to other areas that are bringing information to this area. And then there is the mirror image, which is maybe even in cases in which you have the damage to that specific area, you get a more serious deficit or a more widespread deficit, because that area, maybe the damage to that area is not just preventing that specific computation performed by this area to happen, but it also involving a disruption to the whole network. Okay. And we could discuss about these things for hours, but it's important to bear in mind that these are very important consequences for the whole function. Right. So another important issue in this respect is the so-called disconnection syndromes. Okay. So the idea is that you might have, for instance, certain kind of aphasia, not because Broca's area or Wernicke's area is damaged, but because the arcuate fasciculus, which is this very important tract of white matter of axons, bringing the information back and forth from Broca's to Wernicke's area. Okay, so if you have that like specific white matter damage, then you can have problems, disconnection of the system of Broca's Wernicke's system. Okay, but then again, it's interesting to say that actually people claim that that this kind of aphasia is less severe than Broca's and Wernicke's aphasia. So damage to the white matter alone usually is not that severe because our brain is very good at finding alternative route to transmit the information. Okay. Whereas if you have a damage to the processor, then it's much more difficult to improve with brain plasticity, for instance, or with certain things. Okay. And again, there is also the mirror image of this. So many people say if you have a localized damage to Broca's area or to Wernicke's area, you don't even get a Broca's aphasia or a Wernicke's aphasia. What you need is a damage to the area plus a damage to the surrounding white matter. So if you have a disconnection plus damage to the processor, then you have the serious or the more persistent deficit. Okay. And yeah, so the rule of thumb usually is that the more extended the damage is, the more serious and hard to improve your deficit will be. Okay. And in general, this is also related to the two language areas. So the most serious deficit, aphasic deficit, you would get them when you have a widespread damage to more areas, including inferior frontal gyrus and temporal areas, and especially including the axons as well. Okay. Was this kind of good, understandable, reasonable? Any question related to this point? Okay. Second question, related. Can we really talk about BA44 as an area specialized for linguistic hierarchy construction, considering that in fact, it seems to be highly involved in tool use and a lot of other functions? So this is an issue about multimodality of brain areas, and we are going to talk about that in a few minutes. So thanks for the question. We will discuss it during the slides. Third question. So we discussed the Ding et al. Experiment with the electrocorticography, right? Done to patients with epilepsy undergoing brain surgery. So if the people tested wouldn't have had any sort of neurological condition, would the results have been any different? First of all, we don't really know, right? Second, so what I told you was wrong. These people were not were not undergoing brain surgeries, but epilepsy evaluation. So basically what they did was to record electric currents on their cortex in order to monitor them for epilepsy. And also, especially these kind of electrodes that they would leave in their cortex would be probably would stay there for a few weeks, and then they would record the brain activity in these weeks, and also if they had an episode of epilepsy. However, the assumption is that the brain of epileptic patients or of people who suffer from epilepsy, their normal function is not different. if they don't have an insurgent of an epilepsy attack. Okay, so the assumption is that with respect to main functioning or normal functioning, brains that suffer from epilepsy should not be substantially different from brain of people not suffering from epilepsy, except from the fact that then at some point you have these sudden bursts of electricity, oftentimes traveling from one hemisphere to the other hemisphere, disrupting the functioning of the whole brain, basically, which is the epilepsy. But yeah, good question. Right. So there was an interesting question about the role of the right hemisphere in language, right? And again, we are going to look a little bit into this. Let's say that what it looks is that there is a very strong lateralization for what we might call grammatical relative function. So especially syntax and low-level bottom-up composition processes, compositional related processes, especially with respect to Broca's area, right? So the brain lateralization is strongly found, for instance, in left inferior frontal gyrus. And then the more you move to temporal regions, the more you find bilateral activation. If you remember how hard it was to find a specific activation of the left superior temporal sulcus in the Scott et al. Study, right? So, right. So it's been thought that actually when you move to, for instance, to lexical processing, then you have more of a preference for left lateralization rather than a strong lateralization, okay? But yeah, we, again, as for the specific role of the right hemisphere, it's quite of a mystery. In some experiments, you find an activation of the right hemisphere. In some experiments, you don't find it. A lot of hypotheses have been put forward. Like, for instance, when you have, when you have an increase of complexity of the linguistic material you have to comprehend, then usually you activate the right hemisphere as well, or the right hemisphere has been thought of be involved in like metaphor comprehension or figurative language comprehension, or some specific grammatical operations like binding. If you remember, it's sometimes you get the activation of the right middle frontal gyrus. So yeah, we don't know for sure, but it seems that there are certain operations that recruit the right hemisphere more heavily. Okay. Now, right, now there were like 2 questions about Ding et al. Study, and these are interesting questions. And also I was quite fast when I was talking about that, also because I did not remember that very well. So I reread the paper, and I can tell you something more about that. So the question was, right, so I understood that Ding et al. Found that in their experiment, there was like some structure building happening in the brain. But then why they did the electrocorticography? So why did they implant the electrodes in the brain? And why different electrodes show different activity? But what does this tell us? And how is this relevant? Okay. And related to this point also, if I can give, so they were asking if I can give you some information about the frequency bands, the high gamma band versus low gamma band. And yeah, and also I guess in the ECOG, the high gamma band versus the low, the 124 hertz analysis. What does it mean? Yeah, I haven't talked about that, and maybe I should have. So I'm going to tell you something now. All right, so first of all, the reason why they did electrocorticography was to localize the source, the neural generators of the effect they found with MEG. Okay, so also with MEG, you might have a good spatial localization power, but if you do intracranial recording, you have the highest spatial resolution, because you actually record the electricity on the skull, on the cortex. You plant an electrode within the cortex, basically. Spatially dissociable. Yeah. What does spatially dissociable electrodes mean? So this idea of spatially dissociable electrode means that, so one electrode is recording the neural activity of a small population of neurons, right? And dissociable means that with respect to a certain function, certain electrodes respond and the other don't. So it's a functional double dissociation, right? Their example was phrases, so sentences, phrases, and syllables, right? So they found some electrodes that responded for sentences. So at the sentence rate, which was 1 hertz, but they did not respond for phrases, which was 2 hertz. So it's like, again, it was like, the boy walks fast, right? The boy would be a DP phrase, walks fast, the VP, then everything together a sentence. So yeah, so what does it mean spatially dissociable? That in a specific location where you put the electrode, certain neurons, a neuron population underlying that electrode would specifically respond for a certain stimulus at that specific frequency band. So according to their interpretation, that electrode would be selectively coding that particular representation. So either a sentence or a phrase or a syllable, okay? Right, now a couple of mention to this thing of the gamma band, right? So, yeah. So here they explain this quite nicely. So first of all, they use the English sentence and acoustic control to localize their effect with MEG, right? And with electrocorticography. And the patients were for clinical evaluation, right? Okay, so what is the high gamma band? So the gamma band is basically very fast oscillating rate, so between 70 and 200 oscillations per second, okay? And it highly correlates with multi-unit firing. So the synchronized firing of a lot of neuronal populations, okay? Sorry, of a population of several neurons, a multi-unit firing. And then, so what I didn't explain very well is that, so your question is like, okay, but if we are looking into phenomena that occurs like twice per second at 2 hertz, how can we get them by analyzing a frequency spectrum of 200 hertz, right? So the idea is that what you find at 2 hertz is the modulation of the power of this gamma band, okay? So you can imagine a very quickly vibration, or, you know, a very quickly modulation band, which represent the firing of the neurons, right? And then this sort of very fast vibration modulates slowly at, let's say, 2 hertz or 1 hertz, right? So it's a modulation of a modulation, or a vibration of a vibration, right? So so basically what you have to do is to extract the power band of these frequencies, and then you have a sort of a power spectrum analysis, right? So let's say that you have like a peak of frequencies at 2 hertz, and then if you look at how this frequency changes over time, you're going to find fluctuations of this frequency, okay? It's stupid example, my whistle, right? You remember when we explored the properties of sound. So my whistle might be something that is vibrating at the frequency of I don't know, 4 kilohertz. Okay, so 4,000 of little vibration per second. Now, if I do this, the intensity of this gamma of this frequency band would vary as well. So I'm modulating my frequency band by varying its intensity. Okay, this is in a way what they would find with this analysis. So basically, how many neurons are firing at this rate in a given amount of time, and how this like more firing, less firing, more firing, less firing is distributed over time. Okay. The other analysis I did was the low frequency ECOG waveform. Okay. So again, this is Fourier analysis was directly applied to the ECOG waveform. And then the Fourier coefficients 1, 2, and 4 are extracted. So now you are not extracting the gamma power band. So you're not extracting the power band of the frequencies between 70 and 200 hertz, right? You are just putting the pure ECOG in your statistical analyzer, and then you just extract the 1, 2, and 4 hertz. So you take the electrocorticography as the waveform, the raw electrocorticography waveform as the input, and you just extract these main frequencies. Okay. So one is the modulation of high frequencies. The other one is just the crude extraction of low frequencies. Okay. And the idea is that this low frequency extraction is usually viewed as the dendritic input to a cortical area. So with the first methodology, you find it's more sensitive to multi-unit firing, right? So spiking rate. So when they transmit the output, whereas this low frequency ECOG is more sensitive to the dendritic input, just like the EEG, okay, to a cortical area. So that's why they had these two parallel analyses. And if you remember, they kind of found similar results. So I guess this is a comparison of the high gamma power and the low frequency waveform, right? And you see that this should be sentence. I don't remember. There is no legend. Should be sentence, phrase, and syllable. Okay. I hope. And yeah. And the green dots are electrosensitive to sentence, and the blue dots are electrosensitive to phrases, and these guys are syllable. And yeah, and this would be high gamma power, and this is low frequency waveform. And you see that they're kind of comparable, although you have more significant electrodes for the low. Okay, so again, this is an example of specially dissociable electrode. If you take this blue electrode, the modulation of its signal would be synchronized with the modulation of the phrases. So 2 hertz. So like 2 bumps per second. The green electrode, the modulation of their signal, whether it's like a gamma power band or a slow band extracted from the raw signal, right, would be synchronized to 1 hertz, the sentence rate. Is this clear? is right. Right. Okay. So if this is clear, I would move to where we were left last time, which is which is this. Okay. Okay, so we were discussing some summaries. Can you see the slide? Good. Right. So as a general summary, yeah, we've seen this. We have seen some difference between STS and SDG. I think we kind of got here, right? So this is, again, one of my favorite pictures. It comes from this very nice paper by Binder on Wernicke's area. It's a very short review. If you want to read one paper about aphasia and Wernicke's aphasia, I would warmly recommend you to read that paper. It's super clear. It's super, very well written. And yeah, and that's where he claims, I mean, is quite a solid argument that Wernicke's area has a major role in speech production. Because when you want to say something, you have to retrieve the phonological form of what you want to say. And so its disruption, its damage, brings some problem in speech production, right? And if you remember, this was a very strong counter argument to the idea that Wernicke's area is the language input processing area, and Broca's area is the output, right? To say that this idea, basically the Jesuin Lichtime model, was incorrect, because Wernicke's area is also important for production, okay? And here you see the real Wernicke's area is basically the posterior part of the STG and the supramarginal gyrus. But then, if you really want to have a Wernicke's aphasia, so strong impairment of comprehension, right? You also need a damage to these red areas here, which, according to Binder, contain the word meaning. So the lexical meaning, the conceptual meaning, the meaning of anything related to words. And you see this big blob here, which is basically the angular gyrus and certain parietal temporal junction. It's very important for semantics, and also for concepts, for conceptual knowledge, and also for the interpretation of specific function words, for instance. Function words are known to activate these areas, certain function words, so very important. And then you have the whole middle temporal gyrus and inferior temporal gyrus. Again, lexical meaning, conceptual meaning, you name it, as well as your temporal poles, so your anterior temporal lobe. And here you have the semantic memory of, for instance, single concepts, single individuals, single people, and the something that links the name of, for instance, of nouns, on specific nouns. Okay, we might come back to this later, but the idea is that your semantics, your knowledge about the meaning of words, is distributed along all these areas. And in order to have a severe complete global Wernicke-like comprehension impairment, you need a damage to all this area. Okay. Good. And here it's more about phoneme perception. So this would be more your anterior STG, right? And your, you know, the kind of overacth-like area. So this would be more like speech processing, and this would be more phonological processing, right? And we've seen that. All right. So this is a crazy graph. Usually I would just skip over this and just say, yeah, this is just crazy stuff if you want to read the paper. But actually, now that I'm a little bit wiser and more acknowledged, I want to tell you one thing about this, okay? Just don't read the crazy details. Just focus on this. Here you have what some people call the dorsal stream of language, and here the ventral stream, okay? Now, the idea, according to them, is that, so this is your phonological analysis, again, STG, STS, right? And then this information is passed over either this kind of stream or this stream. Now, here you see this is Broca's area, right? This is your premotor cortex. And this is probably your supramarginal gyrus, right? So this is really kind of Wernicke's area, this and this. But this is what I just showed you as a blob, as a red blob, right? So this big violet thing is kind of this. So it's more your lexical interface and what they call combinatorial network. So certain areas that are deputed of concept composition, right? But you also have your semantic memory here. You also have some of your conceptual knowledge. And yeah, so I don't want to go into details, but the idea is that you have two different streams. This is more grammatically oriented, or as they would say, from sensory to articulatory network. And this is more meaning oriented or lexically oriented, okay? According to this model. Okay. Groczynski's question. Can we build a syntactic map of the brain? Yes, no? We don't know. Some people try to do it. Some people are reluctant for this. But you see that if we just focus on the main areas that are involved in language processing, STG, STS, MTG, left inferior frontal gyrus, middle and superior frontal gyrus, which we haven't talked a lot about, but if you remember, they kind of pop out in certain reviews. So if you just take these areas, then we can just start connecting dots between processes and areas. So take your Broca's area, BA44, active for embedding and XP movement, and also merge. Then if you add BA45, phrase building, XP movement, but also verb level integration. And then you add your superior temporal gyrus. It's also involved in embedding and movement on the left side, whereas on the bilateral side, it's more involved in movement. The other way around, movement is more activating the bilateral STG. Whereas the anterior STG, as Angela Federici said, is more involved in local phrase structures computation, whereas the posterior is more syntactic integration and syntactic operation, a higher level syntactic operation, structure building. Now, right hemisphere, middle frontal gyrus, right hemisphere, binding, left hemisphere, VP movement, superior frontal gyrus, also involved in VP movement, posterior STS, merge and some syntactic integration, left middle temporal gyrus, binding, but also other stuff. So, of course, I don't want you to learn this by heart, especially because this picture might not even be very accurate, right? Just to show you that it's a mess, right? All these areas do a lot of different things. But still, if you are careful and if you read a lot of papers, you're going to find out that actually some of these areas are systematically active in association with certain processes. So if you care about binding, for instance, you're going to find the specific activation of the middle frontal gyrus, which is not usually active for other processes. So the idea is that, according to some people, yes, we can build a neurosyntactic map, right? What this map is going to tell us, it's another question. So this is a nice picture taken from Purple slides. The cartographic imperative, okay? What is that? Is that we have to build a map of the several areas of the brain and localize in these areas all the different functions. But does it make any sense? It tells us a lot about where, about which little dots in your, which little lights in your Christmas tree are activating, but it tells us very little about how it works. Okay. If you find an activation of a certain area, all you can say is that that area correlates with certain process or operation as we posit in theory. But it doesn't tell you whether it is encoding that representation, whether it's monitoring, it's like a higher level modulation effect on different areas, or whether it's just extracting some statistical recurrency out of that phenomenon. Okay, it's really, you know, with correlational data, it is very difficult to infer a causal link. In order to do this, it is better to use causal methodologies like lesional analysis or transcranial magnetic stimulation, where you temporarily knock out very, very small part of your brain or disrupt it. Okay. Why is this causal? Because if from the damage to an area you obtain the damage of a function, then it's a much safer inference to draw the fact that that area was necessary in order for that function to be performed, right? Again, it doesn't tell us if that specific area was housing that function or whether it was a part of a larger network and the disruption of that area was just the problem of disconnection of the network. Okay. Right. So a lot about where, very little about how it works. But we need brain activation maps to have a neuroscientific model of how the brain works in different domains. So it is a starting point. To have these maps, it is a very important starting point. And then if you integrate that map with other data from lesional studies or from other methodologies, then it helps you building up a model of how certain functions work in the brain, right? This, unless you are a dumb neuroscientist who doesn't care about model or hypothesis, right? But let's assume that, you know, as smart people, we tend to be smart neuroscientists. Okay. So, right. Issue about multimodality, and then maybe we can take a break. Okay. So, let's just focus on the most important areas. So, inferior frontal gyrus and superior temporal gyrus and sulcus. Okay. The most important for language. So, just take STS. In STS, you're going to find all this stuff: acoustic processing, phonological, lexical, syntactic integration, and hierarchy. If you move to STG, You're going to find phonetics, syntactic integration, phrase building, movement. If you move to inferior frontal gyrus, Broca's area, movement, merge, syntax semantic integration, phrase building, semantics, the more anteriorly you move, working memory, verbal working memory, action perception. So you see the problem, right? How is it possible that a specific area is doing all these jobs? And how is it possible that one single process is shared by different areas? So again, Pupil has this example that he says that monolithic generalization about function are not even wrong. They're just completely out, okay? What does it mean? To say that Broca's area is the only job of Broca's area is to do syntax, right? Would be completely wrong. We know that is not the case. Areas are multimodal, okay? But what does it mean? Does it mean that there is a specific part of Broca's area which is dedicated to syntax? Or that the whole Broca's area can do different jobs at the same time? Okay. So in this picture, you can see a functional parcellation. This is actually a cytoarchitectonical parcellation of these areas here, right? And you see that what we call BA44 from the cytoarchitecture perspective, so, and also receptor architectural perspective. So this means that if you take the cortex and you look at the layers of cells, if you remember, neocortex has 6 layers of neurons, right? All these layers of neurons have different kind of neurons with different structures, with different orientation, right? Granular cell, stellar cell, pyramidal cells. Certain of these areas are important because they receive the input from other areas. Certain of these areas are important because they retransmit the output, right? Right. So if you look at this structure, you might see that BA44 should be divided in 44D and 44V. So a dorsal part and a ventral part, right? So this means that these two areas are different. And also so their receptor architectonic properties means that what kind of receptors this area use. So, you know, to say BA44 might even be inaccurate by itself. We should always say 44D or 44V, all right? Now, it is true that 44D and 44V are more similar with respect to each other than 44D with BA6, for instance, okay? There is a gradient of similarity within these areas, right? But if you really want to be picky at the level of architecture, we should say that these are two different areas. So if you consider that, it is completely crazy to say that BA44 or the whole Broca's area only does one job, right? You see that, right? Right. And just even if you take BA44, you can also have the functional subdivision. So this is, for instance, a clusterization for 4 different clusters within BA44. And you remember that Zaccarella et al. Identified the peak of sentence structure building versus phrase structure building in one of these clusters, right? Same things holds for STS. Is even worse because STS is like super long, spans through the length of the temporal cortex. And you see, this is a huge review of different studies, finding that in all these little numbers correspond to sensitivity to different functions. So language, non-language, executive, memory, motor, syntactic, emotion, semantic, orthography, multisensory, biological motion, okay? So in your STS, which we heard is important for syntax, phonology, lexical, and semantics, in that very area, you have some parts, some neural population, which are selected for biological motion. You know what biological motion is? When you see some dots moving together, if they move like the joints of a person, you would immediately recognize that, because our brain is tuned to recognize biological motion. And that gives us a very important adaptive advantage of recognizing potential harmful animals or potential eatable animals, right? So yeah, so you might wonder, why the hell our STS is computing visual input and recognizing animals when it's supposed to do phonological analysis or phonetic analysis or syntax, right? So there's no doubt, cortical areas are multifunctional and multimodal, right? In fact, even primary sensorial areas are actually multimodal. They receive afferences from different modalities, right? So, you know, we should be aware of that. Let alone associative areas. So, associative cortex, frontal, temporal, parietal cortex, they are even more multimodal and more multifunctional. Okay, so why? Why are large brain areas multimodal, right? Does any one of you want to try to answer this question? Because I'm a little bit tired of speaking.
Can it be like
What
it's
do you
a
think?
backup plan?
Nice.
It's kind of backup plan if something goes wrong. So I don't know. Or maybe is it that every single stimulus is just very complicated?
Right. So you're saying, you know, it's good to have multimodal and multifunctional areas, because then if one areas get damaged, then you can always use the other one for a different job, right? Or because stimuli are heterogeneous and they are, you know, the idea of a concept requiring a lot of different sensorial, emotional, and motor information, right? Therefore, you need areas that are multifunctional in order to mirror the complexity within the stimulus. Sure. Right. Some other thoughts?
What about, it's very simple and hypothetical, because I don't know what space even means with regard to the brain, but maybe like the brain is finite and there are so many functions, and if one big area would only be capable of like one like, or a little bit like fewer things, then yeah, it would not be enough space in general.
Right. Yeah. So this was actually my first answer. Like, take your temporal cortex. Your temporal cortex is like this piece of brain, right? It's super huge. So maybe it's even not accurate to think about your temporal cortex or your STG as one single area, right? Even if it has similar cytoarchitectonic properties, it's huge. And in this big area, you know how much stuff we can stuff and how many things and how many functions. So yeah, this is certainly one of the reasons, right? So, right. And then if you dive inside, you see that certain little aspect of these areas are more specific for certain processing. Yeah. So all your questions were on, all your answers were on the mark. So maybe if we go quite in detail, very small areas are not inherently multimodal, right? Maybe. Or maybe the problem is not really in the area. Maybe it is not true, for instance, that merge is shared between your posterior STG and your inferior frontal gyrus. Maybe just your inferior frontal gyrus. The problem is that your task is bad, right? And you remember that from Zaccarella's experiment. So he said, look, in all these experiments, find all these activations for composition. Of course, you provide like a real sentence that has a meaning, that even reminds you what you did yesterday, reminds you emotions, triggers images in your brain. That's not going to work. You need a minimum sentence of 2 words. You need to purify your task as much as possible. And this is actually, you know, this is a problem, because it oftentimes that you know, it's frequent that linguists, psycholinguists, and neuroscientists, they say, okay, I found the perfect task to tap on this particular operation. But then if you look at that, then we for instance, we made the example of taking a sentence and scrambling the order of the words and then claiming that is the perfect control without meaning, right? It's not true. That's not the best control there is, right? Because even if you scramble the words of a sentence, you might come up with any crazy meaning that your brain might think of. Okay, right. So as a consequence, neuroscientists today reject the definition of language area. Okay? Many people say that there are no language areas whatsoever. Even Broca's area is not a language area. Wernicke's area is not a language area. What there are distributed networks and distributed representation. Okay? So for instance, this is nice. I took it from a slide of I think of Purple or some conference I went to, and then I couldn't find the original one, and then it was in an Italian article by someone speaking about mirror neurons. So this is supposed to be your brain representation of cinnamon. Okay. What is the meaning of cinnamon in your brain? This. Why? Well, here you have your nice picture of cinnamon. This is the action motor areas that are recruited when you say cinnamon. These are the language areas, so probably your lexical representation of cinnamon, your phonological acoustic representation of someone telling you cinnamon, your memory of cinnamon, the first time that you experienced cinnamon, the last time you experienced cinnamon, the last time you got drunk and you drank tequila with cinnamon. Okay, probably that was not memorized very well, and so on and so forth. Put these things together, that is your concept of cinnamon and your memory of cinnamon. Okay, that's quite cool, and there is a lot of truth to this, right? A lot of truth to this. But then again, I'm gonna come to you and ask you, that's fine, but why? If you have a damage to this area here, your concept of cinnamon will still be there and you're gonna be able to talk about cinnamon. But if you are damaged to this blue dot here, you're not gonna be able to say cinnamon anymore, right? So it is true that these things are distributed and shared, but to a certain extent. And not all of these hubs have the same task. So, you know, in one sentence, the biggest challenge of the neuroscience of the future is to integrate the idea of having a distributed network and of having many different parts of the brain coactive while processing certain stimuli and while performing certain operations, but at the same time, very specific functional localization of representation and processes. Okay, and these two tensions, these are two opposing forces, because functional localization leads to localizing something in a specific circuit, in a specific area, in a specific population of neurons, whereas shared networks and distributed networks go towards the opposite idea, that you need the co-activation of many different neuron population in order to have a function or a behavior. Okay. And, you know, that's another $1 million question. And I would say that we take a little break. Okay. And let's meet in 5 minutes. Again. Yes. Okay, we're back. So okay. Right. Do you see the slides? Good. Right. So, you know, we might end up asking ourselves, is there any functional specialization for language at all, right? If we see that we have this distributed network for processing any cognitive function, especially high-level functions, which require the integration of different lower-level functions, right? Maybe it doesn't make a lot of sense to even speak about functional specialization for language. Now, the fact is that different lesions cause different language deficit. So at some point, we have to account for this selective disruption of function. Now, a possible reply of a very strong distributed network proponent would be that actually, if you have a lesion, then you also might disrupt a specific aspect of a distributed network, and then you might selectively impair the function, right? So, but then again, aren't we talking about the same thing in a way, right? And isn't it all about describing specifically what each single part of the network does, what each single hub or each single processor or each single component, each single computational device, what's its contribution to the functioning of the whole network with respect to a certain cognitive function is. Right. So we know, for instance, that specific lesion can target specific linguistic operations. And we have seen that, right? If you remember the difference in agrammatism and comprehension of relative clauses in English versus Chinese versus German aphasic patients, where the properties of language might interact with the kind of lesion you had. So again, Broca's English patients could not comprehend passive clauses, but then Chinese Broca's aphasia patient could. And the idea is because one requires certain kind of movement crossing the VP and the other one didn't. Right. Another fact is that at the information theory level, at the informational computation, it is really neurons that process information, not axons, okay? So even if you want to put focus on the connectivity of the network, ultimately, the little information processors in your brain, remember, they are neurons, but specifically the interplay of the synapses. So the interplay between receptors and transmitters and the effect caused in the physiology of the neuron due to the transmitters. So it might rise the acceptability or lower the acceptability, right? But then again, this is done at the level of synaptic computation and neural computation. So it's not the axon, it's not the wire itself that processes information. The wire only transmits information. Right. So axonal damage alone cannot explain functional impairment pattern. It has to be integrated with the information about what kind of device it disconnects. As likewise, distributed networks alone cannot explain brain activation patterns, right? If your distributed network is completely holistic, right? It's like it's a sort of a neural network which is just capturing statistical recurrences, then why one part of this network is more selective for certain kind of information, and if that is disrupted, then you're going to have a specific deficit. Okay, that's the big challenge for contemporary neuroscience models. The answer is yes. There is functional specialization for language. Okay, and as it turns out, those areas that are called language areas, they're actually very important for language, and if anything, more important than any area. Okay, because if you have a damage to your visual cortex, you're not going to have the same linguistic deficits if you're going to have any. Okay, right. Now, if we talk about multimodality, yes, Alice, please.
Sorry to interrupt all the time, but
No, please.
are we also going to talk about it in the phylogenetic modality? Like, why is it so? Because is language something that brain was supposed to develop, or from the evolutionary point of view, why is it so? Because I don't know. I don't know anything about this, so I was just wondering whether we're...
Would you like to know something about this?
Yes.
Okay, so I'm gonna mark it and then I will add something about this, okay?
Thank you.
Especially because I'm learning, I'm learning and studying this stuff right now, so you are lucky that I'm sort of, I have this knowledge fresh in my mind, okay? I think they're very interesting things. Yes. Okay. And yeah, so this was a nice sort of illustration that I made to reflect a little bit upon multimodality, okay? And so there are two different hypotheses on multimodality. One is this. This is your neuron or your neural population, right? One neuron or one neural population does 3 jobs at the same time, okay? It is involved in linguistic processing, it's involved in action recognition, it's involved in working memory, the same neuron, okay? So we're not talking about different neuron population within Broca's area, for instance, but we're talking about the same population doing different jobs. So lending its own computational power for different purposes, for different functions. Now, there is an alternative hypothesis to this, which is that that's not true. One neuron only does one job. But it so happens that if you look at Broca's area, for instance, you're going to find some population of neurons residing in the same region doing different jobs. So neuron A takes the money, neuron B cheers up, and neuron 3 drinks beer. There is no such a neuron doing any of these jobs at the same time. You see that this is a different kind of architecture, but at the macro level, you would get exactly the same result, the same effect. That one area lights up for different functions, right? So it is very different to pinpoint these kind of questions, to address these kind of issues. In fact, as far as I know, the only way is to record the activity of a single neuron in order to investigate these questions. And even in that case, it's really difficult to know what exactly this neuron is doing based on what is responding to, what kind of stimulus is responding to. And we're going to have some example of this. So we know that there are neurons like one, but at least some people claim that 2 is definitely the rule. There is a high specificity at the level of single neuron, and it's often the case that multifunctional areas contain different neural populations doing different jobs. Yet, there are some neurons that behave like this multi-job guy, okay? Right. So for instance, the Hailberry neuron, it was responding to visual stimulation, to tactile stimulation, not tactile stimulation, to lexical stimulation, to sounds of Hailberry. It was always lighting up where any stimulus would refer to this actress, right? Regardless of the modality of presentation. So that would be a kind of guy like this. But maybe in a way that neuron would just express the concept of Hailberry and would just receive different inputs from different modalities, right? So it's not an easy question to answer. Right. So we have seen cases of different population of neuron doing two different jobs in the same area. We've just seen it with the Dinger-Hall study, right? Certain population processing sentences, but not syllable or phrases. Other population processing phrases, but not sentences or syllables. Other population processing syllables, okay? So that is an example of neurons within the same area, inferior frontal gyrus, posterior superior temporal gyrus, but yet only sensitive for one particular, let's say, computation or process affecting one certain representation. Okay. And right, so what about neurons that reside in the same region that do different jobs, right? Right, so I'm not sure why lesion study specifically systematic associated with the specific areas. Yeah. So I'm not sure why this is supposed to address this point. Yeah, I mean, I guess the idea was that, for instance, the neurons in the area D would respond for would process this different kind of information. For instance, would be sensitive to like tool use stimulation, to visual kind of stimulation, to memory representation, to tactile kind of stimulation, and integrate all these different kind of certain different kind of information into a sort of a single representation, or into something coding the contribution of the different sensorial areas. But yeah, still, I think this is kind of misleading. Right. Right. So this is a very nice example that I like. So, you know, most of the time people talk about neuron population, but they are very, very fuzzy, vague about telling what exactly the neuron do. And there are some studies, especially in animals, which have recorded the working of a very, very small set of neurons. Like, for instance, this one in the V1 of rodents, okay? So this would be a very, very small portion of the visual cortex of rodents, rats. And what they found is that if you just take this little fragment of the cortex, you would find a modular network organization where the neurons within this fragment would have a lot of synapses between them and fewer synapses with external neurons. So this specific part of the cortex would act as a module, okay? And then the question is there a principle telling us which neurons preferentially speak with? So if two neurons are one close to the other, are going to talk to each other more often than the two neurons that are far away? Or maybe two neurons far away that are processing the same feature of the stimulus are going to communicate more often? What's the answer? The second one. So there is no specific mechanism favoring local clustering, right? whereas the principle is functional specific connection between neurons. So the functional criterion is the principle specifying whether two neurons communicate more often. And what was the functional criterion in this particular case? The orientation of the line that was presented to the rodent. So for instance, take this blue line here with this orientation. This blue, the neuron sensitive to lines of this orientation would be this guy here, and this guy here. Now look at the bodies. One is here and one is here. They are completely on the opposite side of the little fragment of cortex. Still, they would have significantly more synapses among the two of them as compared to this guy, for instance, and this red neuron right next to him. Okay? So at the very, very, very microscopic level, the mechanism favoring communication between neurons is not spatial proximity, but functional proximity. Okay? And this tells us a lot about how neural population are structured. But then again, you know, this is an example of V1. Then you might say, if you move to higher associative areas, things work differently. Who knows, right? Right. So neurons that are close to each other are not necessarily functionally organized, and they're not necessarily, they do not contract more synapses. On the other hand, neurons that are functionally organized, they might be far away from each other, but they contract more, a higher number of synapses. Okay, they're more connected. Right. So functional organization is the rule. For connectivity, not spatial proximity, even at the very micro level. Right. So yes, it is hard to say what specific neural population do, what kind of stimulus they process, how they communicate with other population, how complex representation are shared by neurons. An example, take the elegance effort, a beauty and a beast. This is a worm, a very, very stupid worm. It only has 302 neurons, and we know all of them. It has 7,000 connections between the neurons, and we know all of them. In fact, we know its whole genome. We know all the neurons, all the synapses, and the whole genome, the whole chromosome, the whole DNA, the whole chromosome pool. Good. So do we know everything? Do we have a very, very precise model of how these of how is central nervous system or whatever kind of nervous system it has? No, we don't know anything. It is beyond our understanding. We don't even have the slightest idea of how to model the working of these 302 neurons. Okay. So you might wonder, if we don't even know how to model the nervous system of a microscopic worm, how can we understand how our brain works, right? Yeah. So that's a very nice quote. So like it's fair to say that our understanding of the worm has not been enhanced by having a connectome available to us. We don't have a comprehensive model of the worm nervous system actually produces these behaviors. What we have is sort of a bed on which we can build experiments. And many people have built many elegant experiments on that bed, but the connectome by itself has not explained anything. Okay? So this is very telling about our ignorance about how neurons work. All right, so is neuroscience useful to study language, and how and why, right? Would say the skeptical hippo. And also, as linguists, do we really need neuroscientists to tell us the specific part of our brain create abstract phonological categories, build syntactic structures, and perform linguistic operation on symbolic representations? Do we need them? No, we as linguists don't know that because linguistic evidence is enough. However, yes, and also, after cognitive revolution, linguists became a branch of cognitive science, right? So this is not just a teaser. This is actually what a lot of linguists think, including Chomsky, right? That neuroscience is not advanced enough to tell us anything useful about the brain, right? And how brain computes linguistic phenomena. Right. So we might question.
There was a question by Alice in the chat.
Oh, thank you.
No, it has already been answered. Okay, it's fine.
Okay.
Thank you.
Okay. Are there any other questions related to this? Okay. So what I was trying to tell you is that, you might question what are you doing here, right? Why are we doing this neurolinguistic class if we are not even able to understand how the neuron system of a bloody worm works? So if you embrace minimalist syntax, right, you have two principal operations, merge and copy. And there is no difference from the theory perspective between embedding and movement, between phrases and sentences, okay? But we just saw a lot of evidence that in our brain, different areas are selective for embedding, other areas for movement, and other area for both. Certain neuronal population are selective for phrases, and other for sentences. So this should tell you something about the danger of oversimplifying in linguistic theory. That maybe assume that in syntax you only have two operations is not a great move. Maybe it was better when our theoretical ontology was richer. And maybe experiments on neuroscience and neurolinguistics might tell you, look, you're being too hardcore in your reductionism, in your theoretical reductionism, in your reduction to minimal principles. Maybe it's good to have some richer ontology. And here's the evidence for that. So this would be an example of why these experiments might be useful even for us linguists. I mean, I'm actually not a linguist, but sometimes, I pretend I am. Right. So on the other side, some might say, but look, if you look at the central role of BEA44, right, that actually suggests that probably Chomsky was right. Since BEA44 is active for merge, for copy, for embedding, for movement, for phrases, for sentences, maybe that's where your minimalist syntax is. And maybe the activation of other areas is due to the activation of different kind of information. For instance, of linking syntax to semantic, to phonology, to concept, right? So you see, you have to be very careful. Even if you have some interesting experimental results, you can always look at them from two different perspectives, right? So yeah, food for thought. Right. And then, yes, we know that the different brain regions send back and forth many types of information. We know that we have a lot of feedback, top-down, bottom-up networks. For instance, if you remember, we had seen that, that the phonological mismatch field, right, when you detect the odd phonological category, like the de among all the t's, right? That was popping out in A1 in your primary auditory cortex. But then in another experiment, A1 was found to be sensitive to any kind of sound. So not selective for language, not selective for phonological categories. So that's, if you remember, that was some evidence for the idea that maybe the phonological category was built for instance, in STG, but then this information was retransmitted to your A1 because that's where you're like, that is the phonological change. That is my D among the T's, right? So maybe your conscious experience of the sound B or T is created in A1, but the underlying computation has been performed somewhere else. And you see that I'm switching the order of primary and secondary areas here because STG would be a primary area with respect to phonological processing, whereas A1 would just receive this information after a while. So after having transmitted first to STG and then receiving it back, okay? And if you look at the pattern of activation or oscillation or the interaction between neurons, this kind of feedback circuitry is the rule. It's not exception, okay? So this has to be taken into account. And then how do we know, if you have an active region of your brain, what kind of representation are hosted and what underlying processes are hosted? And that's the source of a huge debate. Right. And also, even if you take lesional studies that are the most precise, that provide the most precise information about a causal link between anatomy and function, even in that case, this link might have been made dirty by the lot of things that happen, like functional takeover. So how do you know if your purest deficit was the strong aphasia that you had when your BA44 was damaged, or your milder form of aphasia, maybe with some mild agrammatism 6 months afterwards? How do you know? It could be that the first hypothesis is right, because that's actually when the real computational device was broken, or it could be that the second hypothesis was right. And then the big damage, the big, the most severe symptom that you had in an acute phase were not just due to the disruption of that area, but the disruption of the whole network or the hypotrophy of the surrounding area, right? Again, we don't know. We might have some hypotheses, we have some ideas, but these things have been debated for a century, okay? Right. So the intact area hosts the functions that were originally hosted by damage, right? But then if as a patient recovered, blind people activate V1, and the hardware is flexible. The brain is plastic, up to a certain extent. So if it is plastic to that extent, what prevents the brain to completely fix itself and show a complete total recovery from damage, right? And then maybe the emphasis should be put on the software rather than the hardware. But then how is it implemented in the brain, the software? Doesn't it lie in the hardware itself? Or yes. So to conclude, in defense of neurolinguistics, the study of the brain, it's difficult, it's a dirty job, but someone has to do it. And we need to start from somewhere. Okay? Second, very nice example. If you need to undergo brain surgery, if there is one thing that the neurosurgeons know is that they must avoid Broca's area. So you might well claim that Broca's area is not a language-specific area. You might well claim that it's a super multimodal area. Still, if you stick a nail into your Broca's area, you're going to have speech arrest and you're not going to be able to talk for a long time. And neurosurgeons knew that. Right. So, a nice way to think about what we're doing is that it actually makes of linguistic theory something that is practically useful. If you ever wondered why is linguistic theory useful, here's an example. Okay, it might tell neuroscientists that actually certain areas are better to be preserved, for instance, under surgery. And also, as I showed you, neurolinguistics can help linguists to refine and rethink their theories, right? And also to evaluate two competing theories, for instance. Of course, it's by no means an easy task. There is a lot of risk of circularity. So stay tuned for the next classes. And yes, the neuroscientific study of language is necessary and it is useful, okay? With all these provisos taken. And then again, Mars can come here to help, and it can give you some ideas on how to come up with some useful theories, how to build bridges from the computational level, more proper to linguistic theory, to the implementational level proper to neuroscience. And the solution is identify the primitives at every level of computation and at every domain. So again, linguistic theory, your primitives could be phonetic features, phonological categories, phrases, hierarchy, movement, argumentative structure. And in neuroscience, your primitives are local representation, distributed representation, sparse distributed representation. Okay? Neural oscillations, rhythms, neurophysiological effects, and all the things we've seen so far. Okay? And this is a nice bubble taken from purple slides. Again, you have an example of linguistic primitive on one side, which can be divided in objects and operations, and neuroscience primitive on the other side. Okay. So, yeah. More questions? What are the primitives in the brain? What is a single neuron? What is a single population processing? How are linguistic representation encoded in the neurons? Via statistical learning mechanism, via different kind of encoding, and what kind of information are transmitted within different regions of the brain, okay? Right, and then are representation really shared? And which features of them are shared, okay? All these are critical questions for building bridges between neuroscience, linguistics. Yes, so in this set of slides, we discussed these papers that I uploaded in the folder. And also I have to thank Andrea Santi and Emanuele Zaccarella that helped me doing these slides. I haven't managed to introduce what I wanted to introduce, but I think we are actually doing quite fine. So we are not late in the program. So I think it was good to take some time to focus on these kind of fundamental issues, and we are not going to come back to these problems so much. Okay.